those who are not vaccinated will end up paying the price.
The Florida Keys Mosquito Control District and British firm biotech Oxitec announced last week that 12,000 of the invasive Aedes aegypti mosquito species are expected to emerge each week for 3 weeks from 6 locations, 2 on Cudjoe Key, 1 on Ramrod Key and 3 on Vaca Key. Eventually it is planned that hundreds of millions of the mosquitoes might be released. Oxitec's non-biting male mosquitoes will mate with the local biting female mosquitoes, and according to the CDC, the genetically modified mosquitoes carry 2 types of genes, a fluorescent marker gene that glows under a special red light and a self-limiting gene that prevents female mosquito offspring from surviving to adulthood. Mosquitoes at the target locations will then be monitored against untreated comparison sites as part of an Environmental Protection Agency-approved project. Oxitec says an evaluation of the project will be provided by the CDC and the University of Florida's Medical Entomology Laboratory, among others.
You are about to enter another dimension, a dimension not only of sight and sound, but of mind, a journey into a wondrous land of imagination. Next stop, the Twilight Zone.
How are you feeling?
Better. How did the test come back?
Your respiratory charts were good. We were concerned about the extent of damage due to inhalation. There were large concentrations of a chemical we determined was luciferin.
Which is what?
The same enzyme we find in fireflies and other bioluminescent insects.
Luciferin is a naturally occurring molecule, meaning it is organic. It can be found in natural species of bioluminescent bacteria, marine life, fungi, and insects such as fireflies. Each bioluminescent organism has a unique luciferin molecule, and they vary in chemical structure and light produced. For example, sea fireflies use their version of luciferin and produce a blue emission, whereas a type of firefly found in North America produces a soft green emission. Some organisms use bioluminescence in various ways, to find mates, ward off predators, and find prey. A bioluminescent millipede uses to show surrounding organisms that it is poisonous and not to eat it. Now, synthetic luciferin has a chemical formula of C11H8N2O3S2 and is a covalent molecule containing nonmetals only. It is clear, opaque, and a liquid at room temperature, similar to water. When luciferin comes into contact with O2 and ATP, it will undergo a chemical reaction. It will then become oxyluciferin, AMP, PPI, CO2, and light. Mg can also be used as catalyst, as shown. It can only happen in a living thing because living things make and use ATP to live. The light is the main importance of luciferin. When creating a light from chemical reactions or releasing energy, lots of heat is produced as well. For example, a light bulb gets hot by the filament when turned on to produce light. So, if too much heat is produced in a living thing, there is risk for injury and death. This is why luciferin is an important and interesting molecule, because of its property to produce light with very little amount of heat. This can occur within a living thing without harm, being non-invasive. With this amazing property, being able to produce light in an organism, scientists can use luciferin in several ways. The primary use is bioluminescent imaging. This has been a very important use of luciferin in human history. With luciferin needing ATP to react, it needs to come into contact with cells of a living thing. Then, when coming into contact, luciferin would react with the cells, and when analyzed through imaging, it can show valuable information, such as a bioindicator to find out if a questionable object is living or not, as well as it can also be used to track cell growth, cancerous cells, tumors, blood flow, infection pathing, and where liquids injected into a living thing go. Here is bioluminescent imaging that is used on a mouse showing cancer tissue and how the brighter than normal tissue around the luciferin is where the cancer is. This is what luciferin has been used in labs for decades to track cell growth, cancer, tumors, and blood flow as said before. Scientists tracked where colored nanoparticles containing luciferin went in the body of mice. As seen in the experiment, the nanoparticles got concentrated in various organs and body parts of the mice. They also used dye to change the exact color of light. Not everything about luciferin is scientific. For example, have you ever wanted a tree or plant to glow? Well, luciferin is possible. Here, a scientist injects luciferin in its liquid state into a plant leaf. Here's another picture of when the leaf is glowing, and then here's another one of another leaf when it's glowing, and this is how it looks like in the bioluminescent imaging as well. Luciferin in plants could replace lights. The plants, when injected with luciferin, can glow overnight. This could be economically efficient and useful, replacing household lights and possibly street lights with trees and plants that can glow at night.
Well, over this decade, we believe unbelievable progress can be made, both inventing new vaccines and making sure they get out to all the children who need them. We could cut the number of children who die every year from about 9 million to half of that if we have success on it. And the benefits there in terms of reducing sickness, reducing the population growth, it really allows the society a chance to take care of itself once you've made that intervention.
Welcome to the mad world we seem to be existing in at the moment. And I don't know about you, but time seems to be speeding up. That's how it feels to me. Weeks go by in a flash, and my email inbox is getting busier and busier with all kinds of news and information, and it's difficult to keep up with it. And I know a lot of you out there, I haven't responded. I do look through all of the emails and I do take it all in, but I haven't got the time to respond to them all. So anyhow, we have this story in Florida. Halt this nightmare. Alarm as Florida set to begin release of genetically engineered mosquitoes. The release of genetically engineered mosquitoes put people in Florida, the environment, and endangered species at risk in the midst of a pandemic. So, what is all of this about? Well, it's only that Bill Gates again. Yes, and here he is. I'm so sick of talking about this guy. This pink jumper wearing boffin nerd who thinks because he has loads of money, more money than sense, he can play at being God. And there isn't anything that this person isn't involved in that isn't about playing God, whether it's buying up nearly all of the farmland in the USA, trying to block out the sun with chemicals, wanting to jab the world with genetic therapy injections, and wanting to create artificial meat and take away all of the cows, and now releasing genetically modified mosquitoes. Through this company, Oxitec, who has invested millions for what it says here to prepare and engage partners for potential self-limited mosquito field trials. And now they are releasing these genetically modified mosquitoes. It says here, environmentalists and Florida residents voiced concern and outrage Monday as state government officials and biotechnology giant Oxitec announced plans to move ahead this week with a pilot project that involves releasing up to 1 billion genetically engineered mosquitoes in Monroe County over a 2-year period. The idea that is claimed is that these male mosquitoes that have been modified won't bite people, but when they mate with the female, they both end up dying afterwards, therefore getting rid of the mosquitoes, which are a problem in Florida because they can carry diseases like yellow fever and such. I mean, oh yeah, that sounds like a great idea. We've got to this point, we've gone through thousands of years, not had to do this before, have we? But oh no, all of a sudden Bill Gates is around and he wants to play at being God. So let's put out 1 billion genetically altered freak mosquitoes and yeah, let's just kill them all off. Another experiment. Sound familiar? Again, it says here, the stated goal of the project is for Oxitec's genetically altered non-biting male mosquitoes to mate with the local biting female population, producing female offspring that die in the larva stage before they can spread disease. A death mechanism. Sounds lovely. Designed into mosquitoes is meant to ensure no viable female offspring will result from the mating. According to Oxitec, the male offspring will pass on the self-limiting gene to half of their offspring, said company spokesman Ross Bethel. Why is this control freak nerd, this boffin with a messiah complex, this unqualified weirdo allowed to do this? Who gave him permission? I mean, does money pay for everything these days? I mean, everything in this world that's living plays an ecological role. It doesn't always have to be positive. It says here the mosquito's purpose in our ecosystem is to provide food and pollination as part of their useful role. The larva of mosquitoes lives in water and provides food or fish and other wildlife, including larger larvae of other species such as dragonflies. So, this guy is messing around with nature again and he's wiping stuff out. And if you start screwing around with nature, it usually finds a way of screwing around with you. And anyhow, people in Florida are not happy about it. It says here, breaking up to 1 billion at Oxitec, GMO mosquitoes are being released for the first time in the Florida Keys. This could negatively impact endangered species and create more virulent mosquitoes. Must halt this nightmare immediately. And here we have in the Daily Mail, Florida residents claim pest control trial that will release up to 1 billion genetically engineered mosquitoes in the Keys to reduce species carrying diseases is terrorism. So what happens to the food chain? These mosquitoes are going to die and get eaten by other creatures? Sounds like another disaster in the making to me. Also, we've seen all of these images from Florida recently with people out and about dancing in bars and restaurants. They have no lockdown, and these pictures are usually used to compare one place with another place that is completely locked down.
Your body isn't well defended against foreign things being stuck directly into your blood. It's a very efficient way around the human immune system.
So this is a talk about gene drives, but I'm going to start by telling you a brief story. 20 years ago, a biologist named Anthony James got obsessed with the idea of making mosquitoes that didn't transmit malaria. It was a great idea, and pretty much a complete failure. For one thing, it turned out to be really hard to make a malaria-resistant mosquito. James managed it finally, just a few years ago, by adding some genes that make it impossible for the malaria parasite to survive inside the mosquito. But that just created another problem. Now that you've got a malaria-resistant mosquito, how do you get it to replace all the malaria-carrying mosquitoes? There were a couple options, but plan A was basically to breed up a bunch of the new genetically engineered mosquitoes, release them into the wild, and hope that they pass on their genes. The problem was that you'd have to release literally 10 times the number of native mosquitoes for that to work. So in a village with 10,000 mosquitoes, you'd release an extra 100,000. As you might guess, this was not a very popular strategy with the villagers. Then, last January, Anthony James got an email from a biologist named Ethan Beer. Beer said that he and his grad student, Valentino Gantz, had stumbled on a tool that could not only guarantee that a particular genetic trait would be inherited, but that would spread incredibly quickly. If they were right, it would basically solve the problem that James had been working on for 20 years. As a test, they engineered 2 mosquitoes to carry the anti-malaria gene and also this new tool, a gene drive, which I'll explain in a minute. Finally, they set it up so that any mosquitoes that inherited the anti-malaria gene wouldn't have the usual white eyes, but would instead have red eyes. That was pretty much just for convenience, so they could tell at a glance which was which. So they took their 2 anti-malarial red-eyed mosquitoes, put them in a box with 30 ordinary white-eyed ones, and let them breed. In 2 generations, those had produced 3,800 grandchildren. That is not the surprising part. This is the surprising part. Given that you started with just 2 red-eyed mosquitoes and 30 white-eyed ones, you'd expect mostly white-eyed descendants. Instead, when James opened the box, all 3,800 mosquitoes had red eyes. When I asked Ethan Beer about this moment, he became so excited that he was literally shouting into the phone. That's because getting only red-eyed mosquitoes violates a rule that is the absolute cornerstone of biology, Mendelian genetics. I'll keep this quick, but Mendelian genetics says that when a male and female mate, their baby inherits half of its DNA from each parent. So if our original mosquito was AA and our new mosquito was AB, where B is the anti-malaria gene, the baby should come out in 4 permutations: AA, AB, AA, BA. Instead, with a new gene drive, they all came out AB. Biologically, that shouldn't even be possible. So what happened? The first thing that happened was the arrival of a gene editing tool known as CRISPR in 2012. Many of you have probably heard about CRISPR, so I'll just say briefly that CRISPR is a tool that allows researchers to edit genes very precisely and easily and quickly. It does this by harnessing a mechanism that already existed in bacteria. Basically, there's a protein that acts like a scissors and cuts the DNA, and there's an RNA molecule that directs the scissors to any point on the genome you want. The result is basically a word processor for genes. You can take an entire gene out, put one in, or even edit just a single letter within a gene. And you can do it in nearly any species. Okay, remember how I said that gene drives originally had 2 problems? The first was that it was hard to engineer a mosquito to be malaria resistant. That's basically gone now, thanks to CRISPR. But the other problem was logistical. How do you get your trait to spread? This is where it gets clever. A couple years ago, a biologist at Harvard named Kevin Esvelt wondered what would happen if you made it so that CRISPR inserted not only your new gene, but also the machinery that does the cutting and pasting. In other words, what if CRISPR also copied and pasted itself? You'd end up with a perpetual motion machine for gene editing. And that's exactly what happened. This CRISPR gene drive that Esvelt created not only guarantees that a trait will get passed on, but if it's used in the germline cells, it will automatically copy and paste your new gene into both chromosomes of every single individual. It's like a global search and replace, or in science terms, it makes a heterozygous trait homozygous. So, what does this mean? For one thing, it means that we have a very powerful, but also somewhat alarming new tool. Up until now, the fact that gene drives didn't work very well was actually kind of a relief. Normally, when we mess around with an organism's genes, we make that thing less evolutionarily fit. So biologists can make all the mutant fruit flies they want without worrying about it. If some escape, natural selection just takes care of them. What's remarkable and powerful and frightening about gene drives is that that will no longer be true. Assuming that your trait does not have a big evolutionary handicap, like a mosquito that can't fly, the CRISPR-based gene drive will spread the change relentlessly until it is in every single individual in the population. Now, it isn't easy to make a gene drive that works that well, but James and Esvelt think that we can. The good news is that this opens the door to some remarkable things. If you put an anti-malarial gene drive in just 1% of Anopheles mosquitoes, the species that transmits malaria, researchers estimate that it would spread to the entire population in a year. So in a year, you could virtually eliminate malaria. In practice, we're still a few years out from being able to do that, but still, 1,000 children a day die of malaria. In a year, that number could be almost 0. The same goes for dengue fever, chikungunya, yellow fever. And it gets better. Say you want to get rid of an invasive species, like get Asian carp out of the Great Lakes. All you have to do is release a gene drive that makes the fish produce only male offspring. In a few generations, there'll be no females left. No more carp. In theory, this means we could restore hundreds of native species that have been pushed to the brink. Okay, that's the good news. This is the bad news. Gene drives are so effective that even an accidental release could change an entire species, and often very quickly. Anthony James took good precautions. He bred his mosquitoes in a biocontainment lab, and he also used a species that's not native to the US, so that even if some did escape, they'd just die off. There'd be nothing for them to mate with. But it's also true that if a dozen Asian carp with the all-male gene drive accidentally got carried from the Great Lakes back to Asia, they could potentially wipe out the native Asian carp population. And that's not so unlikely, given how connected our world is. In fact, it's why we have an invasive species problem. And that's fish. Things like mosquitoes and fruit flies, there's literally no way to contain them. They cross borders and oceans all the time. Okay. The other piece of bad news is that a gene drive might not stay confined to what we call the target species. That's because of gene flow, which is basically a fancy way of saying that neighboring species sometimes interbreed. If that happens, it's possible a gene drive could cross over, like Asian carp could infect some other kind of carp. That's not so bad if your drive just promotes a trait, like eye color. In fact, there's a decent chance that we'll see a wave of very weird fruit flies in the near future. But it could be a disaster if your drive is designed to eliminate the species entirely. The last worrisome thing is that the technology to do this, to genetically engineer an organism and include a gene drive, is something that basically any lab in the world can do. An undergraduate can do it. A talented high schooler with some equipment can do it. Now, I'm guessing that this sounds terrifying. Interestingly, though, nearly every scientist I talked to seemed to think that gene drives were not actually that frightening or dangerous. Partly that's because they believe that scientists will be very cautious and responsible about using them. So far, that's been true. But gene drives also have some actual limitations. So for one thing, they work only in sexually reproducing species, so thank goodness, they can't be used to engineer viruses or bacteria. Also, the trait spreads only with each successive generation. So changing or eliminating a population is practical only if that species has a fast reproductive cycle, like insects or maybe small vertebrates like mice and fish. In elephants or people, it would take centuries for a trait to spread widely enough to matter. Also, even with CRISPR, it's not that easy to engineer a truly devastating trait. Say you wanted to make a fruit fly that feeds on ordinary fruit instead of rotting fruit with the aim of sabotaging American agriculture. First, you'd have to figure out which genes control what the fruit fly wants to eat, which is already a very long and complicated project. Then you'd have to alter those genes to change the fly's behavior to wherever you want it to be, which is an even longer and more complicated project. And it might not even work because the genes that control behavior are complex. So if you're a terrorist and have to choose between starting a grueling basic research program that will require years of meticulous lab work and still might not pan out, or just blowing stuff up, you'll probably choose the latter. This is especially true because, at least in theory, it should be pretty easy to build what's called a reversal drive. That's one that basically overwrites the change made by the first gene drive. So if you don't like the effects of a change, you can just release a second drive that will cancel it out, at least in theory. Okay, so where does this leave us? We now have the ability to change entire species at will. Should we? Are we gods now? I'm not sure I'd say that, but I would say this. First, some very smart people are even now debating how to regulate gene drives. At the same time, some other very smart people are working hard to create safeguards, like gene drives that self-regulate or peter out after a few generations. That's great. But this technology still requires a conversation. And given the nature of gene drives, that conversation has to be global. What if Kenya wants to use a drive, but Tanzania doesn't? Who decides whether to release a gene drive that can fly? I don't have the answer to that question. All we can do going forward, I think, is talk honestly about the risks and benefits, and take responsibility for our choices. By that I mean not just the choice to use a gene drive, but also the choice not to use one. Humans have a tendency to assume that the safest option is to preserve the status quo. But that's not always the case. Gene drives have risks, and those need to be discussed. But malaria exists now and kills 1,000 people a day. To combat it, we spray pesticides that do grave damage to other species, including amphibians and birds. So when you hear about gene drives in the coming months, and trust me, you will be hearing about them, remember that. It can be frightening to act, but sometimes not acting is worse.
How are you going to contain it to the forest? What if the swarm migrates?
The government has initiated eradication procedures. They're quite certain that by using a combination of controlled burns and pesticides, they will be successful.
And if they're not?
That is not an option, Mr. Mulder.
line between disinformation and misinformation is not always an easy one to find.
Governments need to be willing to do things that are out of their historical perspective, or for the most part, it's really a war footing that we need to be on.
It can happen quickly. A Marshall-type plan, I mean, they say that exactly, but a Marshall plan, they can go into effect, can stimulate a change very quickly.
The distrust relates to the health system more generally.
A step up from the part of the government on enforcement actions against fake news.
I think a couple of things we have to consider are, even before this began, the anti-vaccine movement was very strong, and this is something specifically through social media that has spread.
Welcome to Event 201.
Hi, I'm Fox San Antonio's Jessica Headley.
And I'm Ryan Wolf.
Our greatest responsibility is to serve our Treasure Valley communities, the El Paso, Las Cruces communities, Eastern Iowa communities,
Mid-Michigan communities.
We are extremely proud of the quality, balanced journalism that CBS 4 News produces.
But we are concerned about troubling and irresponsible, one-sided news stories plaguing our country.
Plaguing our country.
Cities around the country, including here in Salt Lake City, easing mask requirements, most requiring you have to be vaccinated in order to ditch the mask. Well, ABC4's Jordan Burroughs got public input today around the city. And Jordan, it's all based on the honor system.
Hey, Emily, for the more than 70 people I spoke to, around 80% said they were fully vaccinated against COVID-19 and said they were comfortable sharing that fact. However, on the other hand, there were a bunch of people that told me this needs to be based on more than just the honor system. In 70-degree weather, we asked 70 people some simple questions.
It feels nice to finally be vaccinated and not have to wear that.
Are you fully vaccinated?
I think I would feel more comfortable if either everyone was vaccinated or everyone wore masks until we knew for sure that this was behind us.
This was an unofficial poll of 70 people. 57 say they are fully vaccinated and comfortable sharing that with ABC4. 13 are not, but out of those, only 10 had not received either their first or second round dose. We went around to Liberty Park, the state capitol, and City Creek for that unofficial poll. Father and son Daryl and Nate Luby say they're okay with mask requirements easing, but think local and national leaders need more data and time to make the decision.
I think it's great that we're finally starting to be able to go out without masks, especially outdoors.
We don't really have a way to prove who is vaccinated and who isn't, and I feel like saying you don't have to wear the mask if you've been vaccinated is kind of just an easy way for people who haven't been following the rules to continue not following the rules, and there's no accountability.
What you just said is one of the most insanely idiotic things I have ever heard. At no point in your rambling, incoherent response were you even close to anything that could be considered a rational thought. Everyone in this room is now dumber for having listened to it. I award you no points, and may God have mercy on your soul.
As ABC4 previously reported, the Centers for Disease Control and Prevention eased mask-wearing guidance for fully vaccinated people last Thursday, allowing them to stop wearing masks outdoors, in crowds, and in most indoor settings. Here in Salt Lake City, Mayor Aaron Mendenhall said fully vaccinated visitors will no longer need to wear a mask in Salt Lake City facilities.
Yeah, hey, Wolf, people around the world are celebrating with their cards like this one and looking forward to a post-lockdown life. A lot of people think that countries, companies, and venues may start to require proof of vaccination for people to travel, entertain themselves, or even go to work. That has led to a black market online for fake vaccine cards, as some people want the benefit of the card without actually getting the shot. Now, the major internet sales platforms tell CNBC they're cracking down on people who are trying to sell those fake CDC vaccine cards. eBay told us we are taking significant measures to block or quickly remove items on our marketplace that make false health claims, including vaccine ID cards or similar items. And that could be used to falsely anything that can be used to falsely represent that a person has received the vaccine. Now, Facebook also told us that fake vaccine cards violate the company's policies for sale on its site. To give you a sense of the scale of the problem, eBay says it has removed or blocked 50 million listings now overall that violate its policies around COVID, including price gouging and false medical claims.
If the unvaccinated get vaccinated, they will protect themselves and other unvaccinated people around them. If they do not, states with low vaccination rates may see those rates go up, may see this progress reversed. Ultimately, those who are not vaccinated will end up paying the price.
Hey, Minifish, 17th of May, 2021. I'm just gonna let this play. This is a video that apparently is going viral, and it speaks for itself if you in fact have eyes to see the upside down insanity world that we are quickly careening into. Jesus is the truth, the life, and the way. That's all I can really tell you with absolute certainty. However, I had to just share this with you. This was actually made as a promotional piece for the younger people who are now eligible, if you know what I mean. Pray without ceasing. Keep looking up. God bless each and every one of you. Many fish.
It's vaccination day! It's vaccination day! Today I get my COVID jab, and I'm feeling really fab. For months I have been waiting for this day. When can I get my second shot? And will it hurt or will it not? Oh, I think I am gonna be okay. It's just a small needle, right? And for the people who don't want it and still refuse to take, well, thanks to you now I don't have to wait. 'Cause for the first time in forever, I might get to kiss a girl. It's been a long time. When I get my AstraZeneca, I'm gonna tell the whole damn world, guys, I'm vaccinated! And I know I'm acting crazy, but I mean, I've like waited for a year. And for the first time in forever, I won't live in fear. I won't live in fear. I won't live in fear.
In my administration, we will ensure that this system is in place. And I will tell you, it will be on land, it will be on sea, it will be in air. We will have a proper tracking system.
This is one time, forget the conspiracy. Listen to our government agencies. These guys are telling the truth. You know, there's no conspiracy here, folks. Just get your damn vaccine.
Finally, my brethren, be strong in the Lord, and in the power of his might. Put on the whole armour of God, that ye may be able to stand against the wiles of the devil. For we wrestle not against flesh and blood, but against principalities, against powers, against the rulers of the darkness of this world, against spiritual wickedness in high places. Wherefore take unto you the whole armour of God,